Hello everyone, today I am going to explain details of cell cycle. The cell cycle is a process of DNA duplication and division of cell to produce two daughter cells. That includes many processes for successful self-replication. In 1950s, Almhover and Stephen Park discovered that DNA replication took place only at a specific phase of cell cycle, and this phase was clearly separated from mitosis. Howard and Park worked with the board beam and revealed that the cell goes through the many discrete phases before and after cell division. Understanding with this, scientists identified the four characteristic phases of the cell cycle, such as mitosis, GBE4, DNA synthesis, and GBE4, and understood that in between these phases, cell must also pass through a series of checkpoints. The first step of cell cycle is G1. The first step of cell cycle is G1 step. In this step, cell grows larger, produce RNA, and synthesize necessary protein for DNA replication, and makes more ribosome and organelles. During this period, cell monitors both the external and internal environment to ensure that all the preparation for DNA synthesis have been completed and the overall conditions for cell division are favorable. Cell cycle condition mechanism, which is G1 checkpoint, activated during this period. After passing of G1 checkpoint, cells need to make a copy of its genetic material, so the cell moves into the S phase. Because in this phase DNA is synthesized, the cycle taking approximately 10 to 11 hours in eukaryotic cells, which is taking the longest time in between phases.
In addition to this information, during S phase, cell also duplicates a microtubule organizing structure called centrosome. At the end of the S phase, cell produces two daughter cells. Like the G1 phase, the G2 phase is also a stage when the internal and external environments are monitored to ensure that faithful replication of the DNA has occurred and that conditions are favorable for cytokinesis. There is a major checkpoint at the end of the G2 phase which controls the entire into M phase. The G1, S and the G2 phases together are named as interphase. During M phase, the cell divides its copied nuclear DNA and cytoplasm to form two new cells. M phase is further divided into two phases, mitosis and cytokinesis. In mitosis, the nuclear DNA of the cell condenses into two visible chromosomes and is pulled apart by the mitotic spindle, a specialized structure made out of microtubules. Mitosis takes place in four stages: prophase, metaphase, anaphase, and telophase. Cytokinesis usually begins just as mitosis is ending with a little overlap. During prophase, the duplicated chromosomes condense while outside the nucleus, the mitotic spindle assembles between the two centrosomes. The centrosome is an organelle that serves as the main microtubule organizing center that is involved in the attachment of microtubules to the sister chromatids. During prometaphase, the nuclear membrane breaks apart and the chromosomes can attach the spindle microtubules and begin active movement. During metaphase, the chromosomes are aligned at the equator of the spindle midway between the spindle poles. During anaphase, the sister chromatids synchronously separate to form the two sets of daughter chromosomes. Each sister chromatid is slowly pulled towards the spindle pole it faces. During telophase, the two daughter chromosomes arrive at the spindle poles and decondense. A new nuclear envelope forms around each set of chromosomes, which forms the two new nuclei. This process marks the end of mitosis and sets the stage for cytokinesis. Mitosis and cytokinesis last only about an hour, so approximately 95% of cell cycle is spent in interphase, the period between mitosis. So, how can we tell at what stage an animal cells in the cell cycle?
Is to simply look at living cells with a microscope. A glance at a population of mammalian cells proliferating in cell culture reveals that a fraction of the cells have rounded up and are in mitosis. Others can be observed in the process of cytokinesis. The S-phase cells, however, cannot be detected by simple observation, but there is an accurate method called flow cytometry used for detection of cell proliferation, uses direct measurement of new DNA synthesis. The S-phase cells can be recognized, however, by supplying them with visualizable molecules that are incorporated into newly synthesized DNA, such as thymidine or the artificial thymidine analog BRDU. Cell nuclei that have incorporated thymidine are visualized by autoradiography, whereas those that have incorporated BRDU are visualized by staining with anti-BRDU antibodies. From the proportion of cells in such a population that are labeled, one can estimate the duration of S phase as a fraction of the whole cell cycle duration. Similarly, from the proportion of these cells in mitosis, one can estimate the duration of M phase. In addition, by giving a pulse of thymidine or BRDU and allowing the cells to continue around the cycle for measured lengths of time, one can determine how long it takes for an S-phase cell to progress through G2 into M phase, through M phase into G1, and finally through G1 back into S phase. Another way to assess the stage that a cell has reached in the cell cycle is by measuring its DNA content, which doubles during S phase. Basically, what is the working principle of flow cytometry?
This approach is greatly facilitated by the use of DNA-binding fluorescent dyes and a flow cytometer, which allows large numbers of cells to be analyzed rapidly and automatically. Flow cytometry can be used to determine the lengths of G1, S and G2 phase with M phase by following over time a population of cells that have been pre-selected to be in one particular phase of the cell cycle. DNA content measurements on such a synchronized population of cells reveal how the cells progress through the cycle. Flow cytometry is an extremely powerful technology that allows the individual measurement of physical and chemical characteristics of particles as they pass one by one through a light source. It measures optical and fluorescence characteristics of single cells. Inside the flow cytometer, cells in suspensions are drawn into a stream created by a surrounding sheet of isotonic fluid that creates laminar flow, allowing the cells to pass individually through an interrogation point. At the interrogation point, a beam of monochromatic light, usually from a laser, intersects the cells. Emitted light is given off in all directions and is collected via optics that direct the light to a series of filters and dichroic mirrors that isolate particular wavelength bands. The light signals are detected by photomultiply tubes and digitized for computer analysis. So, the analysis obtained from flow cytometer was used in this study to quantitate the DNA content of stained cells. Thanks for watching.